Assalamualaikum everyone. So today I will tell you what is actually GM crops and the advantage, the disadvantages, and also how GM crops is made. So let's get started. First thing first, GM. GM stands for genetically modified. So, GM crops are plants grown for food whose genes have been altered using genetic engineering. In some cases, the genes of other organisms have been inserted into the genome of the crop plant to improve it. Let's take a look at a few examples of genetically modified crops: cotton, soybean, maize, and canola. Crops have been genetically modified with a gene from a bacterium called Bacillus thuringiensis. This gene causes the cells of the crop plant to produce an insecticide in their leaves itself that kills insects that eat it. The genetic modification reduces the need for chemical pesticides. Rice have been genetically modified to contain beta-carotene, which is used in the body to make vitamin A. This gives the rice a yellow color, so it's known as golden rice. Its taste is not altered, but it contains extra nutritional benefits. Other examples of GM crops include those that grow in difficult conditions, such as droughts or cold temperatures, and crops that are resistant to diseases. All of these genetic modifications, we have only looked at the benefits of growing GM crops. Many people are worried about possible risks associated with genetic engineering. For example, little is known about the long-term effects of genetic engineering as GM crops. Because GM crops haven't yet been around for long, there are concerns that GM crops could be bred with other plants which would spread the modified genes into other populations, and this could have negative impact on whole ecosystem. So, I am going to interview Shirley Settle, a plant transformation specialist. So I would love to ask, can you tell me in general how plant is being genetically engineered or modified in lab itself?
I have some seeds here. I placed these sterile seeds on these plates containing media which contains all the elements necessary for its growth. After 5 days, this is what the germinating seeds look like. Now I'm going to make 7 to 12 horizontal slices. So here I have a bunch of wounded cotyledons. And then here's the agrobacteria that has been resuspended in our co-culture media. And I'm going to pour it over the cotyledons and allow them to sit for about 30 minutes. The bacteria has started to attach to the cotyledons. Now the agrobacteria is attracted by compounds that are generated from wounded plant cells. We move the cotyledons, wounded side down, in contact with the filter paper. The filter paper contains the co-culture media, and we allow these cotyledons to remain on these co-culture plates for 3 days. During those 3 days, the agrobacteria is going to do its thing. It's going to insert its little piece of DNA into the plant cell. After those 3 days of co-culture has passed, we now move these cotyledons to our selection plates. This media contains a compound, a chemical that would normally kill plant cells. We've introduced into these cotyledons with our agrobacteria a new gene that will allow it to grow despite the chemical that would normally kill it. Also in this media is a compound or a hormone that will allow a single cell to grow into a whole new plant. If the single cell contains that new DNA, all the cells in the new plant will contain that new DNA. After 2 weeks, you can see what has happened to the cotyledons. We've started to see a cluster of new shoots being produced. The hormone, or the plant growth regulator, in this media has altered the normal course of development of the cotyledon. So instead of producing a single big plant, we've produced a cluster of buds. You can see here that we have many new plants or new shoots growing from the single cotyledon. And eventually, you will see the start of new shoots growing out of this cluster of shoots. These shoots are our potential transformants.
So that is all from me. Thank you.